class. So today we are going to be learning about NCBI, what it is, and how you can use it to find a specific gene for a certain type of organism, and also how you can find out functional information about that gene within that organism. So we're here at the NCBI homepage, and you can find that at ncbi.nlm.nih.gov. And NCBI, as you can see here, it stands for the National Center for Biotechnology Information. This is a subset of the National Institutes of Health, and they have a wealth of information here on everything ranging from biomedical and health-related literature, peer-reviewed, published in scientific journals. This is a great resource for later on when you're working on your model organism papers. And then they also have DNA, RNA, and protein sequence information that have been uploaded by many scientists from across the world. And they archive that here, and you can look through it and you can sort of get an idea of what your gene of interest looks like and how it interacts with other genes of interest, perhaps. So we're here to look at specific genes, so I'm going to use this drop-down menu to go to Gene, and I am going to type in an example. RAS is what we're going to use for this today. I'm just going to push the search button here. Okay, so here are our search results. We have, in this box here, it's similar to what Google does, where you see, did you mean to search for this sort of thing. And RAS resistance to audiogenic seizures is the main page with the spelled out name for RAS. So you can go in there and you can learn about where it was discovered and a little bit more about it there. And then it also has some suggested organisms that you might be looking for. And then down here is the main list of hits for this gene, and you can see tons of different organisms here in italics, and you can see a bunch of different names, and then they have the locations listed. We're looking for it in a specific organism, and I'm going to look for Homo sapiens, but you might be looking for Mus musculus, which is mice, or you might click into this more or the other taxa in order to find additional organism types in order to study this in your selected organism. These numbers here represent the number of entries for each of these organisms. So Homo sapiens has over 1,000 entries. And immediately looking here, you see that there's nothing that just says RAS. Don't panic. It's common in different organisms for homologs of a gene, which means they are variants of the same gene inherited from a common ancestor of two organisms. Okay, so they're slightly different from one another, but they're similar enough for our purposes. We're going to go ahead and click on the top hit, assuming that that's the best one. It's a fairly safe assumption right now. So immediately in here, you can see that we clicked on the link we wanted, and we can see a summary of the genes, but today we're really interested in figuring out what the gene function is. So we're going to skip over here straight to phenotypes in the table of contents here. Under phenotypes, you can see different diseases that occur when there's a mutation in this gene or in the gene regulatory regions that control its expression. And a lot of these, you can see, are cancer-related diseases. Okay, that's interesting. We'll hold that thought. We'll go back up here to the table of contents, and we're going to go to General Gene Information. So as we look here, we can see some information on gene function. Some of these names are probably unfamiliar to you. We got GDP binding, GTPase activity. Okay, but protein binding is probably something you're a little more familiar with. What else do we have? We have processes down here. Okay, so here I see signaling. There's another signaling pathway. You don't know that, but this is signaling. So this is important in signaling. And what else is very interesting is if you continue on down, it's a positive regulator of cell proliferation, cell senescence, gene expression. This I find interesting because we saw in the phenotype section that this was related to cancer. So this is also something interesting to note, and this tells me that since it's a positive regulator, it's an oncogene, and it's probably upregulated or activated by cancer cells to help them grow faster and survive longer.